rest of the lecture of the principles of crop production. Under the principles of crop production, you have to study about the climate, agroecological region, plant growth analysis, seed physiology, flowering physiology, hormones, and plant propagation. Seed as well as vegetative propagation you have to study. Under that, I think already you have studied about the climate and agroecological regions of Sri Lanka. All the things you have studied, no?
Yes.
Yes. Today we will continue with the plant growth analysis. So before go into the plant growth analysis, we have to know what is plant growth and what is plant development. So what are the phases of growth and types of growth be able to see in the plants. Then we will move to the plant growth analysis. So what is the purpose of doing the plant growth analysis and what are the benefits we are having by go through the plant growth analysis and in which way we can measure the parameters for the plant growth analysis we have to look into. Growth. So that is the permanent or irreversible increase. So if growth happens, it won't change to the normal. So it is permanent or irreversible increase in dry weight, size, mass, or volume of cell or organ or organism. That is the growth. So this is the internal thing be able to see in the living things. So growth is happening in plants as well as in animals. So that is permanent or irreversible increase. That may be the dry weight or size or mass or even it can increase the volume like that. So how we can identify the growth is happening? Because during the growth, cell division take place. The cell numbers also increasing. Cell enlargement take place. So these are the phenomenon even happening in the even plants as well as the living organism. The growth is common for plant as well as animal. So in plant, cell division also happening, number of cells increasing, cell enlargement also happening. So in that case, the growth is quantitative phenomenon because we able to measure that. So that is quantitative phenomenon. With the time, we able to measure the growth. In plant, if you look into the growth, that is generally indeterminate. So plants are capable of growing throughout their life due to the meristematic tissues that is present in the certain parts of the plant that can undergo the cell division, cell enlargement. So this is happening in the plant throughout their life. So from planting to after harvesting, the growth is happening. In plants, we able to see the two types of growth, primary growth as well as secondary growth. Primary growth means the roots and shoot tips, apical meristem of the roots and the shoots that is responsible for the primary growth and elongation of the plant body along the axis. So that is the lengthwise increase we are able to see. So due to the meristematic divisions and enlargement, so of the roots and shoots, so the primary growth is happening along the axis of the plant body. So that case we are calling that as primary growth. Then if you look into the secondary growth, so this is due to the function of the lateral meristem. So the sidewise, the size of the plant is increasing because vascular and core cambium that are responsible for the secondary growth. Due to this secondary growth, the plant girth size is increasing. So the diameter of the plant is increasing. If the lengthwise the plant height that is increasing, that is comes under the primary growth. Secondary growth, due to this, the girth size of the plant is increasing. So that is the main difference between the primary growth and the secondary growth. So this picture is showing regarding the primary growth and the secondary growth. So in primary growth, increase stem and root length. So in axis wise, its length is increasing. So that is primary growth. Here, the secondary growth, a increase in stem and root diameter. The side wise, it is increasing. So that is the secondary growth. Then we will see what are the stages of plant growth, or we can say phases of plant growth. So normally we are able to see three phases of plant growth: meristematic, or we can say formative phase. So due to this, the cell division mainly happens. Then other one, other stage, elongation. So after the cell division, enlargement or elongation of the cell happening. That is the second stage or phase. In third, maturation, some lignification like hardening happening in the plant. So we can divide the phases of plant growth into three: formative or meristematic phase. This time the cell division is happening. Elongation or enlargement phase. That time the divided cell is increasing in size. Then maturation phase. Normally the hardening like liquefaction happens. We will see one by one. So if you look into the formative phase, so it is also can be called as cell formation or the cell division. It occurs at the root apex and the shoot apex, which are having the meristematic tissues. Rate of respiration is very high in the cells, which is undergoing mitosis division during the formative phase. So this meristematic growth can be facilitated by thin cellulosic cell wall along with the plasmodesmata connection. So because the mainly in formative phase, cell division is happening in the root apex and the shoot apex. So if you look into this picture, so this is the shoot apical meristem and this is the root apical meristem. So due to the cell division, this is the number of cells increased. Then if you move to the phase of enlargement, so newly formed cell that is produced in the formative phase, or we can say cell division phase, that is undergoing to enlarge. So due to the enlarging of the cells, plant cells, it develops the vacuoles, and further it is increasing the volume. So due to the formation of vacuoles, the size of the cell also increased. In elongation phase, how can we characterize the elongation phase? Because deposition in the cell wall and increased vacuolation we able to see. So the cell enlargement can occur in all directions with maximum elongation in conducting tissues and fiber, because cell elongation can be happen uh, in all directions. So this is the picture is indicating three phases. So here the formative phase, the most of the cell division because happening in the root apex and the shoot apex it is happening. Then in elongation phase, the formed cells, they are increasing in size. The vacuoles also form. So that is indicated by this, because initially in this size, so due to the other process, it is increasing in size. Then third phase of the growth is maturation phase. Here, the enlarged cells, they develop into special or particular type of cells that is helped undergoing for the structural and physiological differentiation. So this can be characterized by maturation phase of the growth, can be characterized by cell wall thickening and lignification. Cells attain the maturity and their maximal size and undergoes the protoplasmic modification. So this is the thing we able to see in the plant growth. So while growth happening, these are the three stages happening in the plant. So this is indicating about the maturation phase, and these are also indicating three phases. So intercalary meristem also, that is located in the nodes, so that is able to produce the buds and new branches in the plants. Because we are talking about the meristem that is growing. So how it is growing? The intercalary meristem that is in the node of the nodes, so that is produce the buds and new branches. Then we will see, because we are talking about what is growth, then what are the types of growth we able to see in the plant, what are the phases of growth we can see, then we will move to the growth rate. Growth rate, that means increase in growth of the plant per unit time. That can be called as growth rate. So that growth rate may be arithmetic or geometrical explanation. So arithmetic growth indicates the rate of growth is constant, and increase in growth that is occurs in the arithmetic progression like 2, 4, 6, 8 like that. So this is the, if you look into the plant height, so normally we able to get it, the measurements. So this is one of the example for the arithmetic growth. So the arithmetic growth be able to see in the root and the shoot elongation. The lengthwise it is increasing. So geometric growth means this is represented by the initial lag phase, that is slow growth, that is followed by exponential phase of the rapid growth, and the linear growth, the stationary growth, Then again, it is declining growth. So, how the plant is showing their growth performance that can be explained in the geometric growth. So, the growth curve of the plant is one of the examples of the geometric growth, because if you put the seed, so initially the growth rate is slow because only it can utilize the reserve that is available for their germination. After the germination, the few leaves, so that is do the little photosynthesis. So that case the growth is slow. That's why we are saying initial lag phase or the slow growth. Then the plant produce more leaves, seedling produce more leaves. It able to do the more photosynthesis, so growth will happen very quickly. So that case we are saying exponential. So it is increasing with the increasing rate. And after a particular period, the growth is happening, increase with the constant rate. That is the case we are saying that is stationary phase. Thereafter, due to the shading, because lower leaves can shaded by the upper leaves, so light penetration can be affected. So photosynthesis will be affected. So that case, the activity will be, the growth is happening, but is in the slow rate. Then another stage also we are having, if the plant is in the maturity stage, senescence of the leaves happening. So if we draw the curve for dry weight with the time, that is decreasing. Initially, it's showing slow growth, dry weight. Then it's increasing, then linearly increasing, declining, and it's coming to the negative rate. Because so that type of growth we can say geometric growth. The geometric growth is common in unicellular organism, even if you grow that in the nutrient-rich media. Even we can see that in the plant growth curve also. So in geometric growth, so if you look into the dry weight of the plant, so initially the slow growth, then it is increasing, then it is increasing with the constant slope, increase with the same increment. Then it is decreasing. Then in further, if a growth, if you look at the plant growth, it is coming like this. It's further decline. So that is, we are calling that as negative phase. The plant growth is measurable. Anyhow, at cellular level, growth is increase in amount of protoplasm. So this is difficult to measure. But which one we able to measure? We can measure the fresh weight increase or dry weight increase, leaf area, volume, as well as the height. These parameters we able to measure. So to measure the growth of the plant, we can utilize these parameters. So the parameters we are utilizing that can vary with the plant. We can't use the all parameters for the every kind of growth. So depending on the species or the plant, even the organism, we can set. So that's all about the just introduction regarding the growth. So I already told the plant growth is the irreversible change So cell division, cell enlargement take place, that is measurable, that can be measured in length and height in diameter. These parameters can be even leaf area, leaf weight can be utilized. So if you plant the seed, it is little by little, it is growing. So the growth also be able to see. So how the growth is happening? This is happening due to the metabolic processes, anabolic and metabolic process due to the expense of energy. So I already mentioned because the three phases there, cell division, elongation and maturation. Then we will move to the plant development. So the plant development include, because we know what is plant growth, that is irreversible change that is happening in the plant. That two types of plant growth we able to see. There are three stages in the plant growth. The plant growth is measurable one, quantitative one, by using the parameters like dry weight, fresh weight, plant height, volume, diameter, like parameters we can utilize to measure the growth. Then we will move to the development. So the development includes the changes that can happen in the organism throughout their life cycle, from the germination of the seeds to up to the senescence or maturation. So increase in dry matter is the most important parameter we can utilize in the quantitative analysis of the plant growth. So the in plant growth analysis, that is the thing we have to mainly concentrate. Just I want to explain about what is growth and what are the types of growth and phases and development. Then the plant growth analysis is the important thing we have to look into. In plant growth analysis, the measuring increase in dry matter is the one of the important parameter we can utilize. So we can move to the plant growth analysis. So the plant growth analysis, that is the explanatory, holistic, integrative approach we are using to interpret the plant performance and the function. So in plant growth analysis, we have to explain about the differences in plant growth among the species and the same environment, how it grows. Or we can able to see the differences in the plant species, even within the species with the different environment. The different species, same environment, how it's differing, or the different species, the same species with a different environment. Two way we can able to uh, analyze. So even the different species within the same environment, how it's performing, or same species with a different environment, how they are performing. So we can do the analysis. So simply we can say plant growth analysis is the collection of the techniques we are utilizing to describe the plant growth quantitatively, because I already told you plant growth is measurable one. So in which way we can show the plant growth quantitatively. So that to get the quantitative growth of the plant, we are using the collection of techniques to measure that. So that is the plant growth analysis. Then we will see uses or the benefits of plant growth analysis. So by doing the plant growth analysis, what are the benefits we will have? So under that, comparison of growth of different crop species. So different crop species means Even maize is one type of crop, groundnut is another type, paddy is another type. So we can compare the growth of different crop species. So maize is an example for the monocot plant. Then we can say the groundnut is example for the dicot. So what are the difference between the growth of the monocot and dicot? So we can compare them. So that is in that case the plant growth analysis is very useful. Even another benefit or use also having plant growth analysis because we can compare the growth of different crop varieties. Even if we take the cowpea, that is having the different varieties like Bombay, Baruni, Davala, Vijaya. Like that, we are having the cowpea varieties. Some may be in purplish brown color, some may be in reddish color, some may be in white color, some may be in light brown color. So even all are cowpea variety. So that one crop species, the cowpea. Among that, we are having different varieties. So if you want to compare the varietal performance of particular crop, we can go for the plant growth analysis. So varietal evaluation of coffee variety, Bombay, Varuni, Vijaya, Davala, different varieties we are having. Even in paddy, we are having different varieties with a different age, PG 300, PG 350, PG 352, PG 400-1, 406, like that we are having. So that the crop is steady, but among the crop, different varieties. Some may be in red pericarp. Pericarp means inside the grain, if you look into, that is red in color. Some may be white pericarp, white in color. So the within the crop, different varieties also can be compared by using the plant growth analysis. In addition, evaluation of different levels of management that can be practiced for a crop that can also be analyzed because you know if you want to grow the crop, we have to put the seeds and we have to do the management practices. We have to apply fertilizer, water, even weed control, all the things we have to do to obtain the better yield. In agriculture point of view, our aim is to grow the crop to get the maximum yield. That case, the different levels of management we have to give to the crop so that irrigation, fertilizer application, weed control, pest and disease control. So that practices we are saying crop management practices. So if you want to evaluate the different levels of crop management practices given to the crop, how they are influencing, this growth analysis can be utilized. In example, normally Department of Agriculture is having the crop recommendation, fertilizer recommendation for the crop, specific crop. Paddy, they have the recommendation, maize, they have the recommendation. But rather than the recommended level, if you apply more or low, what will happen? That is one kind of management practice we can do. Fertilizer application is one of the crop management practice. Under that, by manipulating the amount or levels of fertilizer we are applying, we able to see the different performance in the crop. So that type of study also we can do, and by using the plant growth analysis, we can measure that and compare the how the levels of management influence the crop yield. So that is also possible, one of the benefits. Then estimation of the net effect of different physiological processes. Normally we know in plants, photosynthesis happening, respiration happening, assimilate translocation happening, plant water and nutrient absorption happening, transpiration happening. So all the physiological processes happening in the plant. So that case, we can even by doing some practices, how this physiological process, we can do the photosynthesis study. Because for the photosynthesis, what are the parameters needed? Normally we need the light is required, then carbon dioxide is required, then water required. So by supplying the different levels of light, how the photosynthesis affected. That is one of the physiological parameter happening in the plants during the daytime. So if we provide different levels of light, how the photosynthesis affected, and this will affect the plant growth in other way. So we can do the estimation of the net effect of different physical processes like photosynthesis. In addition, we can normally we know if we want to grow the crop, different crop need the different climatic condition for their growth. So you already studied what is the climatic parameters that influence the crop production. Even in the agroecological region, based on the rainfall, elevation, and all the things, different regions, the different crops can be cultivated. All the things you have studied. How many agroecological regions you have in Sri Lanka?
24.
46.
46, because the last batch we asked, the 24, that is subdividing like that. That's not the case. The 46 ecological regions we are had. The earlier classification, without giving the A, B, C, D for the regions, so they have the 24. Then further they did the analysis and fine tune the regions. So nowadays we are having in Sri Lanka 46 agroecological regions. Even if we ask in the oral or anything, how many agroecological regions available in Sri Lanka, you will be in a position to answer as 46, not the 20. So that's why just I asked. Okay, because earlier that classification, but later, I think from the 2012 or something, it is 46. So we know the different crops need the different climatic requirement for their better performance. So if you want to test about the particular climatic parameter, temperature or the solar radiation, so we can do that study in the greenhouses by modifying the temperature, because example, for particular crop the optimal temperature requirement is 30 to 32, but if you give the temperature below 30, like 28, and above 32, like 34, so that type of temperature we can provide in the greenhouses and grow the crop. Then we can measure the performance of the crop by doing the plant growth analysis, taking the dry weight or the plant height or the fresh weight or number of branches, like that we can measure. So in that case, the plant growth analysis is helped in the mainly in 5 ways. We can compare the crop in the different environment. Even the same crop, the different varieties can be compared. By providing the different levels of management practices, the growth of the crop can be compared. The physiological process that can happen in the plants can be compared. So how the photosynthesis or the respiration is affected by uh, altering the parameter. So the other climatic parameters also can be modified or altered, and the performance of the crop can be measured by using the plant growth analysis. So now we look into the what is plant growth analysis, what are the benefits we are having by using the plant growth analysis in the crops. So I already mentioned the plant growth is quantifiable. So mainly two basic measurements we are taking to quantify the growth of the plant. The first parameter is dry weight and other parameter is leaf area. These two are commonly we are using to measure the growth of the plant in the growth analysis. So if we want to get the dry weight of the plant, we have to uproot the plant with a particular time interval, 2 weeks interval, because we have to grow the plants in the plot. Based on our needs, we have to uproot the plant from the field. Then we have to get the dry weight of the plant. So to get the dry weight, what we have to do, we have to remove the moisture content available in the plant. So that case, we can use the oven to obtain the dry weight. So if we keep the plant sample at 80 degrees centigrade, normally the range 60 to 80 degrees centigrade we are giving depending on the crop. So commonly 80 degrees centigrade and 48 hours, that is 2 days, we can keep it to obtain the dry weight or until get the constant weight. How we know we get the constant weight? Because we are uprooting the plant and keep it in the oven to remove the moisture content in the plant and get the dry weight. So we have to do this at least 2 days at the 80 degrees centigrade. Then how we can confirm the dry weight is constant? In which way you can confirm that weight is constant? All the moisture that is available in the plant removed, how you can confirm? Any idea? Because at least minimum 2 days we have to keep the plant sample uh, into the oven at 80 degrees centigrade. Then we have to take out and get the dry weight. So we will get some reading. Then again keep the plant sample for some time in the oven at the same condition. Then take the dry weight again. So if both dry weights are same, that is the indication the plant reached the constant dry weight. So if we ask anybody, how can you confirm the dry weight is you are obtaining is constant? So you have to say, because we have to keep 80 degrees centigrade 2 days and get the dry weight. And again we have to keep it in the oven for few hours. Then again get the dry weight and measure. So if two dry weights are same, that is the indication the all the moisture that is available in the plant sample removed. So that is the appropriate way you can get the dry weight. So dry weight is the one of the important parameter we are using to quantify the growth of the plant. Then other parameter is the leaf area. So the leaf area is a total one-sided area of the whole leaf in the canopy. Canopy means all the leaves that is available in the plant and it's forming a structure. So we are calling that as canopy. So while measuring the leaf area, what we have to do in the plant canopy, the one-sided leaf area we have to get it. So the leaf area can be measured in two ways, two methods available to measure the leaf area: destructive method and the non-destructive method. Destructive method means we have to remove the leaf from the plant and bring down that leaf to the laboratory and get the measurement. So that is the destructive. We are detaching the leaf from the plant. So that is the destruction or destructive method. Then non-destructive means while the leaves are attached with the plant, we are taking the measurement. So that is the case we are saying non-destructive, not separating the leaf from the plant. Destructive means we are separating the leaf from the plant. Non-destructive means we are not separating the leaf from the plant. In destructive method, so that can be done in the laboratory, different methods available to measure the leaf area. One method is grid method. By using the graph paper and draw the sketch of the leaf and counting the square, then be able to get the leaf area. That is one method. Then planimeter is also available to measure the leaf area, the destructive method. Then Coke borrow method also available to measure the leaf area in destructive method. But non-destructive method, so the portable green leaf area is available. So that is operated with the battery, charged battery. So we can take that to the field and insert the leaf inside that leaf area meter and pressing that device is there. So by scanning, so by pressing that, that is doing the scan. So here in this picture, you are able to see by pressing this. So this leaf area can be measured. So then we are able to get the direct reading of the leaf area. So the different methods available to measure the leaf area. This is also one of the important parameters we are using in the plant growth analysis, mainly to basic two parameters, dry weight and the leaf area can be utilized. Then we can see two approaches we are able to observe in the plant growth analysis. That is classical approach and the functional approach. So that classification we are having in these two approaches based on the frequency of the sample we are taking and the amount of sample we are taking. So these two approaches we are having classical approach or the functional approach, that is based on the in which interval we are taking the sample. The frequency of the growth measurements are taken and also the size of the plant sample we are taking for the measure. So based on these two approaches we are having, classical approach and the functional approach. So in the classical approach, the measurements we are taking at relatively larger frequencies, nearly 1 month or more than that month frequency we are taking. And that time we have to take the large number of samples. So the frequency is less. So every time we have to take the sample, due to that case, the amount of sample we will take will be reduced. But if we in classical approach, what we are doing every 1 month or 1.5 month interval, we are taking the samples for the analysis. So that case, we can take the large number of samples. So if we use the large number of samples, that is giving the better estimation regarding the biomass and leaf area. But because if you take the 1 month, other reading will be in the second month, another one is the third month. So the sampling points we able to get is the less in the graph. So due to that, the little information only available about the pattern of the growth, especially if we want to see about the growth pattern of the plant. If even if we assume that plant is 4 month duration variety, so if we start sampling 1 month, second month, and third month, then fourth month it is undergo for the harvest. So only 3 points we will get even dry weight versus time graph if we make. So that case, very little information only we able to get from this type of approach, classical approach, because of the lower frequency of the sampling. But in functional approach, what we are doing the sampling very frequently. So 1 week interval or the 2 week interval, we are doing sampling. So that case, so every week we have to take some uprooting the plants to get the dry weight form. So that case, we can't take large amount of plants. So the small amount of sample is only able to get. But in the growth curve, we able to get the greater number of points because every 2 weeks interval, we will get some dry weight. So we will get the point. So that will help to explain, give the information more about the growth performance of the plant. So that functional approach is the very powerful approach compared to the classical approach. So based on the frequency of sampling we are doing and the amount of sample we are taking, two types of classification we are having, classical and functional. In classical, what we are doing, we are taking the sample with a longer duration. If we take the 1 month sample, if we take and put it into the oven for dry weight, then another sample we will get it in the second month and the third month. But depending on the growth duration of the crop, assume that is the annual crop. And what is the meaning of annual crop? Annual crop?
Complete its life cycle within 1 year.
Yes, these plants will complete their life cycle within the 1 year. So that type of example, 4 month or 3 month crop, if we take the sample every 1 month interval, so only if you want to get the dry weight versus time period graph, growth graph, only 2 or 3 points able to get. So it won't give the good information regarding the growth of the plant. So that is the classical approach. But in functional approach, what we are doing, every 1 week interval, we are getting the dry weight. So every week we are removing the plant. So that case the amount of plant we are removing will be less. Otherwise all the plants will quickly uprooted. Or we, in large extent we have to establish the plant for the study. So in functional approach, the time period for the sampling is less. Due to that, we able to get the small amount of sample. But in if you plot the graph, dry weight versus time, we can get the more data points. So the graph will show the each and every growth stages of the plant in their life cycle. So this is the powerful tool. So the two approaches we are having, then we will move to the growth curve because the two approaches we are using mainly to get the growth curve, growth of the crop, we have to study. So that case, the parameters, leaf area and dry weight, the basic parameters commonly used. To get the growth curve, dry weight is commonly used. So the S-shaped curve, if we plot the dry weight versus time, S-shaped curve we able to obtain for the most of the plant species. That is the common shape. So that shape can be called as sigmoidal shape. S-shaped growth we able to obtain for the most of the crops. The common S-shaped graph we can obtain. The S-shaped graph or the curve is called as sigmoidal shape. So variation with the total dry weight with the time is the growth curve. Our aim is to obtain the growth curve. So every time period we have to get the dry weight and plot the graph. And if we get the slope of the growth curve, so that can able to express about the rate of increase of the dry weight with a particular point. If we take the slope, so at that point, the how much of rate of increase happening in the dry weight is indicated by the slope of the growth curve. So if we ask the slope of the growth curve is giving me what? It is giving the growth rate. Normally growth rate we know the how much dry weight increase per unit time is the growth rate we already look into. Arithmetic growth as well as geometric growth already studied. So in plant that is showing the geometric growth, the S-shape growth that can be called as sigmoidal shape. If we take the slope of the plant growth curve, that will give the growth rate of the plant. So that is giving the crop growth rate. So that is the thing we will get it in the growth curve. So to understand the physiological basis of the plants in the growth analysis, we can further divide this sigmoidal S-shaped graph into 5 phases. So I will take that, I think, in the next class. Lag phase, exponential phase, linear phase, decline phase, and negative phase. I will take that in the next class. So today we started to study about the plant growth analysis. So if we want to study about the plant growth analysis, we have to know about what is plant growth, what is plant development. So growth is the reversible change that is happening in the plant, that is measurable one. The growth can be happen in 2 types: primary growth, secondary growth. And while growth happening, 3 stages we able to see in the plant: cell division phase, cell enlargement, and maturation phase. Then growth rate is how much of increase we able to obtain in a particular time period is the growth rate. arithmetic growth and geometric growth is available. If you look into the plant growth curve, that is the sigmoidal shape, A shape, that is the geometric plant curve growth rate. Then we move to the plant growth analysis. The plant growth analysis is a collection of techniques we are using to measure the plant growth quantitatively. By doing the plant growth analysis, it's having the benefits. Be able to compare the different crop species, even be able to compare the growth performance of the same crop of the different varieties, how the level of management practices influence the growth, and climatic parameters influence the growth, and the physiological parameters, how can be influenced by adjusting the growing environment. So these type of benefits we are having by doing the plant growth analysis. And also in plant growth analysis, two basic measurements we will take. Dry weight and leaf area is commonly used. Dry weight, we can keep the plant sample into the oven for 2 days at 80 degrees centigrade and get. To leaf area, there are two methods mainly available, destructive and non-destructive method available to measure the leaf area. In two approaches, we are utilizing to measure the plant growth, classical approach and the functional approach. Depending on the how much of sample we are taking and the duration we are utilizing the sample, it can be classified into classical and functional. In classical, we are taking the sample very long duration. So due to that, we can able to get more sample. So it is not accurate because only 2 or 3 points we able to get if you plot the graph. But in functional approach, the frequency is very less. Every 1 week we can get the sample, so less amount of sample can be taken. Otherwise, it will finish the sample, the plants that is available in the field. So we can, if we plot the dry weight versus time in the functional approach, be able to get the more points and get the graph neatly. So it will explain about the each growth stages of the plant. Then plant growth curve. In plant, be able to obtain the S-shaped curve. If you plot the dry weight from seedling to up to the maturity, if we take the dry weight of the plant, be able to get the S-shaped graph, that is sigmoidal curve. That can be further divided into different stages, 5 stages. We will study that in the next class. So lag phase, exponential phase, linear phase, decline phase and the negative phase, and each phase we will study a little deeper in the next class. I will stop after this today. Then next Thursday, I will continue the lectures. Thank you. If you have any doubt, you can clarify before starting the next lecture. You can clarify with the next part. Thank you.
Thank you, madam.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.